So today we're going to talk a little bit more about using Samplicons to do samples to do Amplicon analysis in KBase. So we're going to start off with talking a little bit about uploading Amplicon data. I'm going to give you a brief overview here on sort of the importance of metadata and how we're integrating that across DOE systems. And then we're going to walk you through an example analysis workflow. Kathleen's going to take the wheel on that and show you some of our tools for doing data manipulations, taxonomy assignments, ordinations of community data, categorical stats, SIMPR analysis, and a functional prediction. Kathleen is a postdoc at Argonne who's been very generously working with me on developing these things. She's research-wise, she's interested in plant-associated microbial communities, but she's been involved in the development of the Amplicon analysis functionality in KBase, as well as the community standard for Amplicon reporting formats I'm going to be talking about today. And as been mentioned, I'm a computational ecologist at Argonne, and I've sort of been leading both of those efforts. So here's the broad overview slide of how all of these data are integrating on systems using samples. So you have your sample metadata, and we're using that to do analysis of various different data types that we're allowing upload to system. Today we're going to be focusing on the OTUs, and OTU is broad here to include any taxonomic unit, including ASVs, from Amplicon data. So we'll be walking a little through this data itself, as well as that the analysis pipeline. So the Amplicon reporting format is something that has been developed in conjunction between KBase and ESS Dive with feedback from the broader DOE microbiome community. This is a live standard, which means it's open for continued feedback from other people. We are in the final stages of preparing the documentations for ESS Dive, and then it will be available for comment and suggestions on their GitHub page. So that's to happen in the near future. But the purpose of this is that a lot of microbial data has been collected, and it's very difficult to evaluate what data you could use in a meta-analysis or pull data together across different studies, different labs. And so this standard supports FAIR data principles. We've developed it to be easy to use, so we're not asking super hard questions. A lot of the fields are optional, but the more you provide, the fairer your data are, the more easily other people can use your data again. And that's really important as we're moving towards more open science and ability to ask bigger, broader questions by pulling together existing knowledge from other data sets. We've really focused on supporting data reuse and interoperability. We designed this across different platforms so that there would be sort of an ease of use and you can transfer data across systems. That was very important to us. And also, we really tried to focus on the metadata that was most critical for understanding whether or not you can use two different data sets together. So that is really the focus. Right now, you'll see that you have to enter the data into KBase directly, but the plan is once APIs and everything get going with the different projects, you'll be able to seamlessly share data across platforms. So if you bring data into KBase and use it there, you'll be able to then push all of that data together as a package to the ESS Dive repository if you're a DOE researcher, because they only support DOE funded projects at this point. Or if you've already put your data into the ESS Dive repository, be able to pull that into KBase. And that functionality would help people who have their data in ESS Dive, as well as people who are interested in analyzing data off of ESS Dive. So that functionality isn't here yet, but that's definitely something that we're discussing and have plans to develop here in the near-term future. So the reporting format consists of 4 different files, which seems like a lot, but it's different kinds of data, right? So when you're doing your Amplicon analysis, you know you have your giant reads files, and so that gets sort of collapsed in the standard down to a FASTA file that has a single representative or consensus sequence for each taxon, however you defined taxon in your own taxonomic abundance matrix. And this matrix is stored as sort of a flat file, so a comma delimited or text delimited file, with the row being the taxon and the column being the sample. Right, and so that's just a file that contains the raw count data for each of your sample by taxon table. Then there's a separate file for the sample processing metadata. So this is different than what we're going to be talking about today, the sample metadata on system, which is about your collection metadata. This is about how you did your sequencing, basically. So what technology and platform did you use? What target gene your gene was? Kathleen will go through this in a little bit more detail when she's talking about the Amplicon upload step as well. And then there's also a bioinformatic processing metadata, which covers your clustering method, quality filtering steps, so sort of what you've done after you got that data onto the computer. Here in KBase, these two, the sample processing metadata and bioinformatic processing metadata, are both manually entered at upload. All right, so instead of having two files, you just manually enter it into this somewhat overwhelming at first glance, but we promise it's not actually that hard to do. And as Kat walks you through it, it won't be too hard, you'll see. Format when you're doing the upload here. So today we're going to talk about some of the cool analyses that this allows you to do in system. So once you have uploaded your Amplicon matrix, you'll be able to assign taxonomy using the RDP classifier. We have different training set options for that. You can view the taxonomic distributions in bar plots, in addition to the visualization that comes along with the RDP classifier. You can do matrix transformation. So like I said, upload your raw count data, and then you can do your abundance filtering on taxa or samples, any sort of normalization, relative abundance, transformations that you want to do on that. You can do it on system, and then that tracks those changes that you've made and stores it in provenance on system. So again, that helps with the data reuse and interoperability. Then we have some apps that enable you to do sort of basic categorical statistics is what we're going to cover today. So Anisim, Permanova, looking at sort of the dispersion of your data using categorical variables, as well as doing a simple analysis. So which taxa are really driving the differences in composition among those categories. And then predicting community functional potential. So these apps are still not quite ready for full release, so they're in beta mode, but they'll be fully released soon. In addition to these, we have a few other options that are already on system, including Mantel tests, making co-occurrence networks, and doing correlation analysis, but those will be presented in later webinars, so we're not going to present on those today. Okay, and so with that, I'll tell you a little bit about the data and then hand it over to Kathleen to do the actual walkthrough of all of these apps, showing you the configurations and how to do it on your own data. The data set that we're going to be using for this today was collected as part of the Argonne Wetlands Hydrobiogeochemistry SFA, which is a DOE-funded project. And this particular data set that we've uploaded is looking at the influence of microtopographic position. So is it a higher or hummock position versus a low or hollow position on soil microbial communities beneath two different plant communities. So we have a pine community and a red maple community. So with that, Kathleen, I'll go ahead and stop sharing screen and you can start with the walkthrough.
Sounds good. All right, my screen should be visible now. Is that true?
Yep. Yes, looks great.
All right, great. Okay, so I'm going to walk you through some of the nuts and bolts of how we use apps to analyze the data stored in an Amplicon matrix. So to start with, we need to get all of our data into KBase. And so we'll use this import samples app here to upload data on the environmental material from which our samples were collected, from which our Amplicon sequences were generated. And this app was covered in the previous samples webinar, and it's also covered in the KBase docs. Both of those are linked through the Q&A document for this webinar, as Ben mentioned at the top. And so once we've run this app, we'll have uploaded that sample metadata. And we can see here that we've got a table and we have some sample IDs, names that are specific to the study. And then as we scroll across, we can see some of those different variables that Pamela mentioned were in the data set, which includes the physiographic feature, whether it's a hollow or a hummock, as well as the locality, the pine backwater or the floodplain. So you can view that once you've uploaded the samples. All right, so the second part of this sample upload, of this data upload process is going to be importing the Amplicon matrix. So for that, we're going to use this app to import the Amplicon matrix. And so the parameters here, first, we're going to have a sample set reference. So this is where we're going to choose the sample set that we just uploaded containing our metadata. And then we'll come down here, and this is where we're going to upload our count table. So this is going to be a tab separated variable file, that tab separated value file that's going to contain the samples as columns and the taxa that we're looking at as rows. And it's going to have the counts for each taxon in each sample. And we're going to upload this from our staging area. And so if you come over here to the data pane and click this little arrow and go to the import tab, this is where you would drag and drop your files from your computer or upload from the web to get the Amplicon count table into the staging area. And once it's in there, it will be available to you in this drop-down menu, so you can select your Amplicon table. And then the second part of this is that we're going to need the representative sequences for each of your taxonomic units in that table. And so for that, again, we're going to get a drop-down menu here where we can go to the staging area and select a FASTA file that we've uploaded containing those sequences. All right, so now brace yourselves. I'm going to click this Show Advanced Parameters. And at this point, we're going to see all of the metadata fields that are available for you to fill out in association with this Amplicon matrix. And it is a bit overwhelming at first, but really there are two basic types of metadata we're looking for here. The first type is kind of your sample processing metadata. And so that's going to deal with whatever you did to go from your sample to sequencing reads. And to the extent possible, we've named these to correspond with Mixis fields. So you've got the opportunity to provide some links to describe your extraction and amplification process. And then at the very minimum, in order for the uploader to work, we want you to tell us what marker gene you're looking at with these Amplicons and which subfragments. So in this case, we would select V4. Sorry, you also have the opportunity to enter things like your primers, your library kit layout, any screening that was done to the library. And if you click on each of these, you can see a short description of what we're looking for there, where the sequencing was done. So that's kind of the first set of the metadata, the processing metadata. The second type of metadata we're looking for is bioinformatic metadata. So this is going to be the metadata that you used to go from reads to your Amplicon count table. And so whatever you did in order to prune those reads to a particular length, pair them if that was necessary, a certain error rate that you were willing to tolerate when using your barcodes to assign those reads to samples. And most importantly here, we want you to tell us whether you used a denoising or a clustering based method to process those reads into OTUs. And so in this case, we would say clustering, and you have a free field here. So even if you've used a really custom pipeline, you can tell us a little bit about what method you used to do that and what your cutoff was in terms of sequence similarity for defining OTUs. And so that's an overview of the metadata we're looking for. And once you've filled that out, you can give your new Amplicon matrix a name and click run on this app, and then the importer will run. And once it's run, We'll see here, we'll get some basic statistics on the matrix and a little heat map there. And I'll scroll down here. And so now this Amplicon matrix is available to us over here in the data pane. All right, so at this point, before we jump into analysis, you may want to perform some pre-processing on your matrix. So you can come to this transform matrix app and you can select the Amplicon matrix. And then you can, in this first part, define a series of operations you want to perform. So for example, you may want to do some abundance filtering. You may want to say, I want to drop samples that don't have at least a certain number of total reads. Another operation we may want to perform after that is to transform our matrix to relative abundances for some of the analyses that we're going to do downstream. All right, so I've defined the series of operations that I want to do. And then at that point, brace yourselves again. We're going to click show advanced here. And now we get a lot of options to define what we want to do in each of these operations we've selected. So for perform abundance filtering, I may want to say, let's remove columns that don't have at least 10,000 counts across all of my taxa. And then I can select which way I want to, whether I want to calculate that relative abundance on rows or columns. In this case, since we're using sample. Oh, sorry.
But
Can
I
you hear
think
me now?
we just lost about 15 seconds of you. But
Okay, what was
you
the
now,
last
yeah.
thing I said?
Yeah, you're just talking about performing abundance filtering.
All right, so yeah, I can just tell it what the actual threshold is that I want. I only want to keep columns that have a sum here of 10,000 at least across all of my taxa. And then when I'm doing relative abundance, I can say I want to do that on the columns, the relative abundance of the taxa within each sample. And then I can click run on that app. And when it's done, I'll provide a name here for my transformed matrix. And so I'll be able to identify it when it appears here in the data pane. All right, so now that we've uploaded our Amplicon matrix and done some pre-processing, we want to use that Amplicon matrix and our sample metadata to do some analysis. And so we kind of have 3 analysis use cases that we're going to explore here. And so the first thing we want to do is to assign taxonomy to our OTUs and visualize the taxonomic composition of the communities in different types of samples. So to do that, we're going to start by using this classify rRNA with taxonomy app. And so this is going to run RDP Classifier, so we can select our Amplicon matrix. We can give a name for the output matrix that's going to have the taxonomy assignments. And then we can select which classifier we want to use. In this case, it's for 16S, and define a confidence threshold below which we do not want to include taxonomy assignments for the Amplicons. And once we run that, we will get this output, which we can explore if this opens. There we go. All right. So now we can come into this pie chart and explore which species we found in different clades within this data set. So I can check for my favorite taxa and make sure that they're all here. All right. So now that we've assigned that taxonomy, we want to select the taxonomy that we just built. Now, for a given Amplicon matrix, you may want to assign taxonomy using different thresholds or using different classifiers, and all of those will be available to you here, so you can select the one you want to use. And then if we click show advanced here, what this will do is it will give us access to that sample metadata that we have linked to the Amplicon matrix. And so I can say, I actually want to generate this bar plot and organize the bar plots by those physiographic features, so that I can compare them. And once we run that, here's an example of the output we will get. Oops. All right, so you can see here we have these stacked bar plots representing the taxa that are present in each of our samples. And those bar plots are organized by which type of environment they came from. So we can very quickly scan across and try to see if there are any interesting patterns there. All right, so the next use case that I want to describe deals with looking for associations between the composition of these communities and the environmental factors that we've recorded in our sample metadata. And so there are three approaches we're going to look at here. The first is NMDS. So that's non-metric multi-dimensional scaling, and we're going to ordinate our samples using this approach. And this runs the meta MDS from the vegan software package. And so we're going to select the Amplicon matrix. And we can select whether we want to perform this on rows or columns. So in this case, we want to look at those taxa in the rows and compare our samples based on their composition with respect to these rows. We can enter parameters for the number of components and the number of iterations. And then if we select these column attributes from our Amplicon matrix, what that's going to do is when we select group marker by, we now have access again to that linked sample data. And so I can come through here and say, you know what, when I ordinate, I would like you to give me some colored groups that are based on physiographic feature. So then we can give that a name, run the app, and here's an example of the output. The samples are ordinated. I'm going to try to select this app cell so that it's a little clearer. So you can see the samples are ordinated here based on a dissimilarity matrix that was calculated from the Amplicon matrix, and they're colored based on an environmental factor. And as a second example of that, here are our samples again, colored factor that may be of interest for further analysis.
Kathleen,
okay,
sorry,
so the second approach we can use to look for these
we lost you for just about 15 seconds there when you were explaining the PCOA plot. Sorry. If you just want to start from the top of that.
Yeah. All right. So just from the beginning.
You could just do the second one. Yeah. Just
okay.
do one of them.
Yeah. All right. So this is the second plot. And so here we have the samples ordinated and they're colored in blue for the hollow and in red for the hummock environment. And we can start to see some separation between these groups. And so this points us towards this environmental factor being important with respect to community composition. So the next approach that we can use is to look at that by performing a statistical test to see whether a distance or dissimilarity matrix produced from that Amplicon sample is significantly associated with one of those environmental vectors. And so to do that, we can select our Amplicon matrix again, and again, we can select the column attributes here. And what that will do is it will make it accessible for us here to select one element of our sample metadata that we want to perform this statistical test on. So for example, I can select the physiographic feature name here to define the sample categories. And then I can choose what type of distance or dissimilarity I want to calculate from the Amplicon matrix. So select columns. In this, I'm interested in testing differences between sample groups. So those are our columns. And then we can enter a number of permutations and which tests we want to perform. And so when we hit run, we can get the results of that. And so for example, here we have the results of this ANOSEM test showing us that in fact, there was a significant association between that environmental variable and the dissimilarities between our sample groups, representing their community composition. All right. So another type of analysis we can do is this similarity percentage analysis using SIMPR. And so this is going to allow us to examine which taxa seem to be contributing most to those dissimilarities between samples. So to do this, again, we select the Amplicon matrix. We select the column attributes, and we can use our sample data to define the groups that we want to look at. All right, and then when we run that, we'll get the results in this tab showing us the OTU names for our most influential taxa. And we'll also get this nice bar plot, which will be broken up by the environmental variable that I selected from the linked sample data. And this will show us the average abundance for a given OTU here along the x-axis in samples from those two different types of environments. Okay, so the final analysis use case that I want to show here is the prediction of functional potential of a community based on Amplicon data. And so to do that, we're going to run this app that uses PyCrust. All right, and so that's phylogenetic investigation of communities by reconstruction of unobserved states. And so we're going to select the Amplicon matrix. We can select which annotations, which types of functional annotations we want to take a look at. We can enter a name for the output, and we can run that. And we'll be able to download the tables that show us the functions in each that were predicted for each Amplicon or for the total community in each sample. And we can also view some heat maps to kind of scan over that data. All right, so that's the walkthrough of some of what you can do with your Amplicon matrix in KBase.
Super, thanks so much, Kathleen. And yeah, I guess we'll pass it back to Pamela here.
We have just a couple other quick slides here. So basically, it was just to remind you of the upcoming related webinars on samples and chemical abundance, which I think Ben has linked the registration link to in the Q&A document. And then after that, we'll talk about using samples to link multiple forms of measurement and performing the analysis across them. So that's where we'll get into some of the other apps I mentioned earlier, like the Mantel tests and the ability to do sort of the correlation and as opposed in continuous variable analysis, as opposed to the categorical analysis that we showed here today. And then I wanted to give an acknowledgement to the team. This was a big group effort, and you know these apps were developed by a lot of people and did a lot of troubleshooting. So Kathleen and I did a lot of the testing and sort of thinking through what we wanted to see. We're open and excited to hear what functionality you guys would like to have on system. So if you can include that in the Q&A or contact us separately for that, that would be great. And with that, we'll wrap it up, and I'll hand it to Ben if he has any other closing comments.
Yeah, super. Thanks, Pamela. I just posted a link as well in the chat to our samples survey. If you haven't filled that out, please take a look at that and fill it out so we can, you know, like Kathleen or like Pamela was saying, we can, you know, better understand the use cases that you're trying to use these apps and new tools for. So thanks again, everybody. We'll, you know, keep these the Q&A document open there. If folks still have any questions, we can answer those for the next hour or so. And we will be posting this up on YouTube. So if you want to, you know, rewatch any of Kathleen's walkthrough of the apps, you can do that. So thanks again, and sign up for our next one. It's going to be on December 8th. I just posted the link in the chat there, and it is up on our website as well. All right, thanks everybody, and have a great rest of your day.
Thanks for watching this webinar. For more webinars and tutorials, subscribe to our YouTube channel. You can see announcements for upcoming webinars and recordings for previous webinars on our website at kbase.us/learn. Let us know in the comments what content you'd like to see in future webinars. Be sure to follow us on Twitter at DOE-KBase. And if you have questions or encounter an error when using KBase, please contact the help desk.